So Jens Hauser is a Paris-based media studies scholar, writer, and art curator, focusing on the interactions between art and technology. He has recently been a professor in art history at the Karlsruhe Institute of Technology and currently is researcher at University of Copenhagen's Medical Museum, as well as distinguished faculty member of the Department of Art History and Design at Michigan State University, where he co-directs the Bridge Artist in Residency program. At the intersection of art history and epistemology, he developed a theory of biomediality as part of his doctorate at Ruhr University, Bochum. He also holds a degree in scientific journalism from University France. Okay, thank you. As a curator, he has organized around 30 international exhibitions and festivals, including at least 2 editions together with RIXC.
Thanks a lot, Rasa, for this kind introduction and overall also the renewed invitation to come back to Riga, which is a place that has been providing a lot of challenges and pleasure over the last decades, I would say. And whenever I try to fit into the context, of course, I look into how you phrase the questions of mediality this year. It's a media art festival in a certain way. And the notion of media, of course, mutate as organisms do. So when looking at the program of symbiotic senses, you aim to embrace symbiotic thinking, I quote, fostering collective action towards a more symbiotic future. Symbiosis, a concept originally coming from biology, denotes a collaboration among different species, shaping the course of evolution. And you also quote Lynn Margulis' pioneering work on symbiotic systems to contemporary discussion on topics such as extended cognition, holobionts, interconnectedness across species, and so on. And the idea posits that you write, the symbiosis defines our existence and highlighting our complete interdependence on our environment and planetary system. So with my humble contribution today, entitled Art and the Holobiont, Microperformative Positions Beyond the Individual Collective Binary, I will try to both take the challenge and locate these questions within the given context of media art, and in the light also of a forthcoming book, Life is Other, Abiotic Entanglements in Art and Creating, that has just gone to print last week, and that I have co-edited with Thomas Feuerstein and Lucy Strecker, following 2 exhibitions in Austria. So the concept fosters transdisciplinary discussions in art philosophy and the technoscience in order to destabilize and blur the boundaries between self and other, biological and technical realms, hence contribution to shifting discussions from identity, individuality, difference to cohabitation and entanglement. And I will therefore elaborate on how the concept of the holobiont proposed by biologist Lynn Margulis inspires performative and microperformative artistic practices. The festival's title, Symbiotic Senses, implies that sensory perception may not be just a feature of individual organisms, but a symbiotic collaboration, and triggers the question of what is a medium today, enabling sensing and measuring, beyond the still typical device, function, or infrastructure-based view that just technical media represent, control, transmit, store, or process information. So let's imagine that we ask some cell biologists instead of the typical media art scholars to bring their medium. And they will most likely not come up in company with a visionary shaman. They could. It's a medium as well, right? Neither with a television or a tablet computer, but maybe with a thermocycler, kind of molecular photocopying machine, or even more likely with a flask of gross medium for cell culture. That means that the meaning of the concept of mediality itself mutates and needs to be seen historically in its etymological and epistemological evolution. As historian Erich Porath reminds us, in the past, the word medium was more frequently used in the concept of the natural than of the human science as intermediary element, as milieu, and only much later as tool or means. And for this reason, today's dominant focus on the functions of communication causes us to lose sight of the natural scientific relevance of mediality, so that, quote, cultural studies and traditional media studies generally lack consideration of the natural science and the history. I think that is what you have to try to change with this festival. So, accordingly, to analyze strategies of so-called wet media art, I developed the theory of biomediality and tried to set up a grid of three main types. First, life-enabling milieus, biological media to be understood as existential conditions that enable a body and its internal functions. Technically, they can comprise experimental systems where milieus are simulated biotic or abiotic alike. For example, in tissue culture, where incubators or growth media are necessary. So the second would be the very notion of media that we're all familiar with, technical means, so maybe biomedia as well as transformative, generative means, whereby biological systems do something, but beyond their original organic purpose. So they are processing bodies, molecules, organisms, populations, cells as synthesis factories, viral promoters, or programmed bacteria, for example. And then the third one, instances of measurement, media of biology employed to measure, and this is maybe the festival topic, measure, analyze, and observe. So displays in which one organic system reveals something about another to produce knowledge, and historically positioned in the tradition of microscopy or cell cinematography, such as biomarkers, DNA chips, biosensors, or even gel electrophoresis. So a paradox appears, and which is important with regards to biomediality's conceptual link to agency, performativity, and co-corporality, because the usual claim that media need to be stable, dead, neutral, or soft transmitters of hard content, supposed to be indifferent vis-à-vis what they control, transmit, store, or process, here fails, because in the case of biomediality, media do not only produce what they pretend to merely mediate, they have their own agency. For example, biological media in the sense of milieu are in the focus of the Victimless Leather Project by the Tissue Culture and Art Project, where they grow miniature leather-like jackets out of immortalized animal and human cell lines. And they use this liquid growth medium that contains fetal calf medium. And this acts as a growth stimulator. And while the product ordered online just seems to be a standardized substrate, it indeed consists of an overlooked cocktail of multiple microperformative agencies. But often researchers even ignore the exact composition of such media. So it appears to be passive. In fact, it contains signaling molecules that activate cell growth and trigger intercellular changes. The second example would be biomedia, and here in a biological sense, of course, transformative media implying to modify organisms such as to accomplish technically programmed features. For example, Joe Davis' bacterial radio uses variants of a gene from orange Marian puffball sponges in transgenic bacteria that are here employed to plate electronic circuits and thus to grow a crystal radio circuit. However, the bacteria may get contaminated or die, and so may the radio. Media of biology, this is what we are interested in, maybe in this festival mostly, employed to measure, can also be seen in Paul Vanus's performative gel electrophoresis installations, which subvert so-called genetic fingerprinting to create figurative motifs instead of the abstract, usually appearing band patterns. And the artist employs enzymes to cut genetic sequences, primers, and molecules, probes to produce these DNA images. However, the calculated motifs are never clean digital images. They're only approximate and full of smear, since manifold biological agents involved depend on a large range of parameters that condition their life and work. And then you can also measure in an ecological sense. You may know this project by Brendan Ballengee. In the first step, he tried to have all this kind of milieu effects on a frog extinct since years, and trying to rebreed against the timeline a frog that has been extinct due to human pollution, and to breed against the timeline. And here we have the milieu influences on the frog and rebreeding generation to generation to actually come closer. But we have the frog as a medium, as a historical trope, as we can see in Gavani's experiments with bioelectricity, and using the bioelectricity of a frog in order to measure the bioelectricity of another organs, for example, a fish. And we can here, for example, use, or Brendan Ballengee uses in this project MALAM, so malformations in amphibians, to actually use frogs as ecosensors, to say, are there too many predators? What about chemical and substrates pollution and so on? And to actually use populations, not just individual frogs, but populations of frog as an ecological indicator for the healthiness of the environment. There's some other measurings where, of course, we have the choice between, is it a kind of individual or is it collective sensing? And this is an interesting example which we just discussed in a seminar I was happy to attend, where Rasa SMITE also presented her own research about how a plant may sense another plant. So by the simulation of a green filter, a green light, the proximity of another plant individual is simulated, so to say, to test whether there is a kind of change in growth. So to say that it's a decision making how to grow, that depends on how the competition or the plant itself is on competition. And here's images that Rasa is happy to share with me also, because she presented this last week in a seminar on plant intelligence in Basel. So it's interesting question to ask whether it's the individual that also senses the others, or how far the plant itself is already connected to the root systems, to other communication systems, to root communication, to not only to light sensing, but maybe also to VOCs and so on. So the question of whether individual or collective is maybe very tricky here as well. And after all, isn't the plant also already a collective, in the sense that all the chloroplasts are, according to the symbiosis hypothesis by Lynn Margulis, already encapsulated prokaryotic organisms that, as chloroplasts, function here as a kind of proxy of endosymbiosis. The same applies in a little bit to quorum sensing. And quorum sensing in bacteria, for example, relies on chemical signaling molecules called autoinducers, which regulate bacterial gene expression. And like languages between humans, these signals vary between species. And every bacterium senses changes in the concentration of the autoinducers in the surroundings. And sensing a sudden increase in autoinducer concentration changes the bacterium's gene expression, protein synthesis, and behavior. It will adapt to group behavior, while a bacterium that senses a drop in autoinducers will adapt to individual behavior, and bacteria optimize their behaviors to work efficiently in a group. So also we have this kind of the concentration of the beyond the boundary, but the boundary, the membrane, is also permeable, so that the autoinducers can pass. So this kind of binaries are very tricky, because on very different levels, we have this functional kind of definitions. It depends very much on which level you locate the individual versus the collective. And here I don't want to go into details, but we go from complex functional space-time systems on the basic level, biochemical, biophysical, then a kind of physiological evolution, the development of cell organelles, multicellularity, differentiation, up to nervous systems, tissues, organs for locomotion, orientation, reproduction, and so on. And then the next level of ecological, social functions, symbolic communication, and transmission systems. And this, of course, reminds us the very notion of individuation. Individuation not only in the psychological sense, such as popularized by Carl Jung, where individuation is meant to describe a natural process of a development of that is unique to an individual, and human, of course, self-understood, to becoming a whole by separating oneself from the collective. But even more in the theory of Simondon, in a kind of more biological centered approach, such as by Simondon, individuals in the philosophical sense are neither considered to be ontologically indivisible, and indivisible is what individual means, but in the way that the living becomes indivisible. So it's a becoming, not the being. That is, the living is modifying its relation to the environment by modifying itself. This brings us, of course, to the understanding of biological individuality that is challenged by the concept of the Holobiont, a term coined by Lynn Margulis in 1991, or taken up and developed symbiosis as a source of evolutionary innovation to define the theory of symbiosis linked to miniature ecosystems. So it's claiming that eukaryotic organisms are not autonomous individuals, rather we are holobionts. And in the recent years, the Holobiont concept has emerged as a theoretical and experimental framework to study the interaction between hosts and the associated and microbial communities of all types of ecosystems. So we have organized 2 exhibitions about the Holobiont then, which laid the ground for this forthcoming book. And this needs to be seen in the context of the social and psychological transformations triggered by the COVID pandemic. And the evidence that life is first and foremost that of a large variety in agencies other than human. So we experience us as transitory beings drifting between digital and molecular worlds and sense the twisting of boundaries within us as the emerging potential and the possibility of a biosymbiotic understanding beyond a symbolic distance from the world with an increased potential for multisensory entanglements. So the question we asked were how many biological entities, machines, media, architectures, networks develop symbiotically in the context of contemporary biological possibilities and ecological challenges, and how do the arts transpose this shift, prompting in turn new challenges for staging and curating, which is the subtitle of the book. So the concept of the Holobiont comes, of course, from the tradition of holism, a school of thought advocated since Aristotle that considers physical, biological, social, and political elements in a system as interconnected. An increasing number of artists today focus on this overlooked interconnections, rather on the reductionist sides of the first generation of so-called bioart, focusing on either genetics or tissue culture, but having a kind of hybrid practice, and to look at these various components and emergent properties. So their position differ from another trend in the digital arts, in the media arts, at large, namely to negotiate personal human identities as constructed. For example, queerness is very often thought as a social political phenomenon against biological determinism, which makes sense, right? Or alluding to nature's queerness, mainly as an argument for human social role models, but without thinking with the biological other. So against this background, we wanted to have an alternative to say, well, we'll look at otherness, and it's not about human self-identity and teenager crisis, to make it very sharp. So overcoming the temptation, for example, to merely update transhistorical traditions of artistic self-portraiture within an even larger variety of media in times where selfies have become a global phenomenon of human individual identity construction, artists raise new questions about binaries such as self and other, difference and sameness, individuality and collectivity, or carbon and silicon-based worlds. Of course, we have Arthur Rimbaud's famous 19th century declaration, "Je est un autre," I is another, right? Which has been interpreted through various lenses, including Lacanian psychoanalysis, since it addresses one's fragmented self, suggested that there is a creative impulse to be imputed to psychological splits or external forces beyond the artist's genius and cognitive control. But today, however, this other tends to be even physiologically explainable, and indeed based on the natural science, for example, with a discussion about the gut-brain axis, and can be addressed in terms of multi-species alliances. What if, for example, we considered, as writes Doreen Sagan is Lynn Margulis' son and also Carl Sagan's son, and his contributor to our book, and also a long-term collaborator with her mother. So he writes in our book, "Consciousness, my I-ness, is just a job that the consciousness of my 90% bacterial cells and the obligate 10% animal multicellular cells require me to do, in other words, our planet mates. The other through space we share and whose shared species and spaces indeed we are." And taking this argument even further to the origins of humans' sensory nervous system, media philosopher Bruce Clark argues that whatever has evolved over time into today's nerve cells, allowing for perception, thought, speculation, and memory, are nothing else than large-scale manifestations of the small-scale community ecology. This is the fusion of two ancient forms of bacteria. We'll come back to that the current nerve cells in evolution terms, according to Margulis, are the result of prokaryotic cells joining in a certain moment in space and time. So emphasizing such symbiotic relationships challenges human ego concepts and more broadly existent orthodox notions of defining biological individuals. So this is exactly this term, this term of Holobiont that is rather than Darwinist notions of competition between organisms through reproductive fitness, is referring to the concept of symbiogenesis. For example, that organelles, mitochondria, for example, or chloroplast, they were just once independent bacteria, and now they're included into eukaryotic cells. And now the concept, of course, is extending to other fields. It's interesting that while the first staging of Holobiont Life is Other, there's a kind of lapsus, and you will see this. We actually named the two exhibitions differently, once with a V and one with a F. It was a bit of unconscious, but then we thought there is a reason, kind of Lacanian, kind of acte manqué, or a lapsus révélateur in a certain way. While the first staging of Holobiont Life is Other in Bregenz relied entirely on works that could be digitally transmitted rather than physically shipped, the second version, after the sanitary restriction had been lifted and even crossing borders that were not problematic for the virus but problematic for us, integrated then a number of material artworks and performative installations, including various biological agents. So the exhibitions condensed bodies, environments, media, machines, organisms into multimedia spaces, each of which engaging with a narrative about another life and about the lives of others, from which the glyph that we designed, kind of, I think we have seen it here, the glyph, which fuses the F and the V, is coming from. So it has been designed for this publication. And the LF likewise expresses life's multiplicity, as all other lives at the same time alludes to extension of genres of performative or life arts, time-based and media arts to increasingly include not only human bodies, but a much broader spectrum of microperformativity. So accordingly, Holobiont Life is Other was framed by a large mural wall newspaper of our complete special issue on microperformativity, which I can pass around here if you want to have a look. There are also some flyers for the forthcoming book, since the book is only available in December. It was encouraging visitors to climb up, to get a ladder, to get a chair, to climb up on stools, crawl in unusual positions on the floors, or to take any non-central position in order to get in touch with these holobiontic narratives, cognitively as well as bodily. And even the more the question of the adequate media and mediation got into the focus of how to create microperformativity and otherness. Of course, audiovisual documentation of performative actions, physical remnants, or digital 3D animations are established standards. And in our case, the exhibitions both also were made as well accessible via a 360 degrees virtual Matterport tour. But concepts of mediality mutated in the light of collaborations between artists and scientists. We asked, for example, what would a biological like that we always are familiar with in the digital space, what could we have as a transposition into the biological realm? What would a biological like resemble? And our answer was this closed reading station proposed by David Barry and Lucy Strecker, and it invited visitors to select and comment on passages from the journal by speaking into what looks like a microphone, but which however does not record speech, but the reader's breath and the microbes it contains. So it's a display reminding Thomas Feuerstein's microbograph from 2002 already, where he modified a Hasselblad camera, replacing light exposure from aerial exposure, freely adapted from Henry Flex Tolbo Pencil of Nature. So it's in German, it's a Belichtungszeit that becomes a Belüftungszeit for those who get the pun. So the publication Life is Other takes the exhibition Holobiont as Other as a starting point in combining theoretical approaches and deepening discussions departing from the concept of microperformativity, which is a neologism I've coined over the last 20 years in order to denote this current trend in theories of performance and performativity and performative artistic practices to destabilize human scales, both spatial and temporal, as the dominant plane of reference and to emphasize biological and technological micro-agencies that beyond the mesoscopic human body that we act in, relate to the invisibility of the microscopic and the incomprehensibility of the macroscopic. So the special issue for the journal Performance Research on Microperformativity from 2020 during the viral pandemic resulted in 27 international contributions by 44 contributors. And we choose Bleu Remix by Geneva-based performance artist Jan Marusic as a cover image for our issues for two reasons. First, because it subverts the tradition within which human phenomenological considerations are still rooted by displacing the focus from the mesoscopic actions to microscopic functions, from physical gestures to physiological processes, and from stage diegetic time to real performative time. In this mesmerizing physiological performance, the former dancer, getting rid of and tired of dancing, Marusic staged a controlled internal biochemical choreography of methylene blue, which progressively seeps out of all his orifices in the body, from eyes, mouth, nose to eventually the pores of the skin, while internal bodily sounds are amplified and remixed, shifting the observer's attention from the physical to the physiological space. Another, in a similar vein, Spela Petric's 20-hour-long performance Scopopoesis can be conceived of as a microperformative physiological encounter between human life and vegetal otherness as a co-performer, adjusting their respective rhythms and functions. And here the artist and germinating crest stand vis-a-vis each other, illuminated by a strong artificial light. And the title Scotopoesis literally means shade by darkness and refers to the continuous shadow of the artist's body that signals the plants to respond by changing the shape and color so that their bodies in turn produce a living imprint of the artist's silhouette. Or the deconstruction of apparent stillness also was at the core of the unsplit micro-performance and macro-metas festival created at the Muffathalle in Munich, Germany. And for example, here dendrites are growing in real time and out of vaporized mantle on the performance body in crystal, a project by Russian artist Julia Borovaya. While in Mars Bloom explorations by Tina Taubgott, a human performer interacts with thousands of mealworm non-human performers sought to slow down audiences to get in touch with decay, decomposition, and death. And under this dome, multiple cycles of life create constant changes on the landscape, while mealworms digesting polystyrene, human waste, and critique a unique soundscape. A very known example, of course, of human relationship is May the Horse Live in Me by French duo A Orienté Objet, where artist Marine Lavaljonté was injected with compatibilized horse blood to experience immune otherness in an act of transspecies blood or sisterhood. We have compiled a list of micro-agencies that play a role, and just to read it through, it's quite amazing which kind of elements are being isolated and can be acted. But of course, it's more interesting to have these coming together than to stage this in isolation. So with Chrysaltas Bonmot, we went further in the construction of the book as well. Bacteria perform processes, scientists perform experiences, algorithms perform actions, humans perform gender and sex. The question is who or what doesn't perform nowadays today. And this is a very short flick through the actual book. So the authors that we have, Monika Backe, Thomas Feuerstein, Bruce Clark, Eduardo Katz, Paul Vanus, Klaus Spies, myself, and Maja Smreka. Just a very short glimpse. Of course, we introduce all the possibly microperformative entanglements in our introduction chapter. Then in Monika Backe's chapter, we actually put a clear-cut abiotic distinction to the test of a deep time biological and geological perspective of metabolic forces, and speaking with Karen Barat of a kind of metabolic-based evolution of mineral species. Thomas Feuerstein insists that we have to move from metaphors to metabols, so to read actually the very processes and to have the media, the material media, be in the focus of the reading, so have a kind of epistemological understanding of what the current metabolic functions are to turn the metaphor into a metabol. And very interestingly, and I mentioned this in the beginning, As a known connoisseur of Lynn Margulis' work on symbiosis in the Holobiont, Bruce Clark focuses on one of the main poster protists for symbiogenesis in order to speculate on humans' ability to speculate and the biological basis for cognitive capacities in general, to depend on the progressive evolution of a rare consortium called Mixotricha paradoxa, in which a protistian eukaryote is mixed up with radically different strains of bacteria. So it's like the assumed role is to be the origins of our all sensory nervous systems today, to make us see that in the biosphere altogether, complex life persists through symbiosis all the way up to Gaia as a planetary system and back down to microbial dark matter. Eduardo Katz's pieces, this kind of biotopes, are also a very interesting case of collaboration, as are Paul Van Nuys's installations, where he's recreating the 3 different strains of bacteria fulfilling work. But here we have the smell of sweat not as a byproduct, but as an end product, and no human is working anymore. And the microagency of the 3 different bacteria produce the smell of the human labor and eliminating the human labor altogether. I want to go just very quickly. This is Klaus Spiess's attempt to extend the linguistic concept of speech act towards its ultimate materialization by spotlighting oral microbes as co-host of human speech that push back against the traditional notion of language as exclusively human domain. So it's actually building a gym for microbes, so to adapt through phonetic stimulation the microbiome and to re-enrich the microbiome that we are all losing because we are now all speaking this airport English, and also diminishing the biodiversity of our oral microbiome. In this performance I created with Maja Smreka in Berlin some years ago, it's about hormonal stimulation. So Maja Smreka is raising a puppet with a puppy without being pregnant, and actually stimulating not only her lactating mechanisms, her breast milk production, but also the stimulation of hormones such as serotonin and oxytocin, and to become mother and to feel the motherness with otherness. And this project has been awarded the Golden Ica also at Ars Electronica, when she then went on to actually extract ovary cells from her cells and to replace the actual nucleus of the cells with the nucleus from a dog, to be deep frozen and cryoconserved for our species future, when we have more empathy towards more kind of trans-species families in a possible or impossible future. And then I just want to close with this interesting article by Doron Sagan. So as I said, Carl Sagan, the Nobel Prize winner, and Lynn Margulis were his parents. So it was just amazing to have him as a contributor to this book. And he is considering from a broad cosmic evolutionary perspective, we are the epiphenomena as humans of a multi-billion-year-old and perhaps cosmically distributed, highly durable and intensely social microorganisms. And outstanding artists, he claims, may have possessed a genius that was partly precipitated by spirochetes. The artists were, in a sense, more than human. So Sagan's provocative call here is in order to broaden up the view of art, which includes all life as well as a human artist, with or without illusions of personal grandeur. And to finish with, for our exhibition Holobiont Life is Other at Bregenz, Sagan and Clark have contributed to the artistic rendering of Margulis' theories entitled Planetary Holobiont 2.0, comprising a particularly striking example of a holobiont that you may know of. I'm speaking about the case of endosymbiosis in this Hawaiian bobtail squid, which is home to the bioluminescent bacteria Vibrio fischeri and feeds them sugars and amino acids, while the latter become a special light organ to the squid's mantle. By the symbiosis here, the squid can match the ambient light of the night sky and hide the squid's silhouette when viewed from below, and this counter-illumination protects it from predators. So could there be a more romantic story of symbiotic entanglement than that of a marine animal escaping its predators in the moonshine thanks to prokaryotic otherness in its belly? So this is the end of my story to tell, but hopefully the beginning of just much more others to come. Thank you.
I didn't want to take up space, but nobody's raising a hand, so I have this one idea which crossed me, and I wanted your comment on that, Jens. So how is your notion of microperformativity with all these agents on all these different scales? How is it, I'll do it like this. How is it different from the object-oriented ontology approach? You know, where you have also an infinity of scales, where there's all kinds of agents. And my sort of very blunt notion of that is that your microperformativity is alive throughout. And the object-oriented ontology idea is pretty dead.
Yeah, I think it's a very interesting comparison and analysis. I think that the departure of what these artistic practices have induced is mostly based on biotic, and the object-oriented one more on physics and maybe matter. But there's a combination of both that I think with especially Monica Bacchus' contribution to the emergence of mineral species, and where mineral species often been former plant life, that this distinction between the biotic and the abiotic have to fail. But there's a lot of flat ontology kind of parallels to object-oriented ontology. And I'm, of course, obviously a good friend of Tim Martin. So, yeah.
I have just maybe one question. So, okay, you refer to symbiotic systems, you refer to Lynn Margulis, and the artworks, however you use. So basically, for me, it seems like you are kind of either mixing up or intentionally or switching from to both Darwinism and the symbiotic, like innovation and evolutionary theory, with all the neo-Darwinists against which Lynn Margulis was very strongly, which is this line of all this gene modification biotechnologies. So, and you are completely confused, making this switch between both, and yet contextualizing culture, of course, and all these discourses. So, what you can have to say about it? Human and non-human.
Well, in the examples that you saw, very often there is an anchor point, which is a human performer interacting with other agencies, which is, of course, a way in for many spectators and all the audiences to just need to kind of this anchoring point, because just bacteria and pathogens are not always very sexy and appealing. I mean, aesthetically, it's a problem to transpose these thoughts into something that speaks still to an audience. This is one point. And second, I think that we still are trapped by binaries such as Lamarckism against Darwinism. I think that if Darwin would have met Lamarck, they wouldn't have so much disagreed. It would be nuances, but we are making a black and white scheme of that. A lot of writing has been produced in the last 20 years around neo-Lamarckism, also in the light of post-genetic, epigenetic approaches, the regulation of the gene rather than gene, the regulation and the interpretation rather than the information. And this is what was always interesting to me in Nordic and Estonian and also Danish biosemiotics, is to shift from information theory to interpretation. I think this is something that Jesper Hoffmeyer, as a biosemiotician, has also told me in his writings, that it's not relevant what is informed in a gene, because what is really counting is how it's interpreted. And to shifting from information to interpretation also, I think, is cutting short the discussion about whether there is or not a kind of Darwinian predominance, or whether the regulation factors are dominant, right? So I think that we are still fighting in these terms. I mean, this kind of binary that we as a kind of dialectic device need in order to make sense. But I think, the boundaries tend to blur even between these lines of thought.
Yeah, okay, thank you, but maybe more questions from the audience? Just my last comment is about, so that... Anyway, the gene engineer is kind of, they can claim that they are, so that evolution will anyway make changes. Well, but that's such a big scale. So that basically, if you do this gene engineering, you don't give a space for the living bodies, living organisms to adapt in a more, maybe more relevant way, let's say. Yeah. So no, I'm just thinking because Margulis was following Darwin very much, but she was against neo-Darwinism. And that just for the audience, just to make this clarification also that today, because we are in this context, because I'm now working with organic farming, also research institute in Switzerland. And because we have had for long discussions for these couple of years about, so where is these boundaries? So until which they are breeders, yeah, so they are growing crops which are could be also adaptable and could survive climate change and these radical conditions. But also, so where is this point where they should stop and not anymore interfere into the gene level? So they usually try to stop in a cellular level because this is the level until which evolution has been going and naturally and helping these organisms to survive and adapt. Yes, but here what we see in most of your cases in art, so this is just what I wanted to say, that it's a total, like kind of, you use the theories of following Darwin, following symbiotics, following of all ideas of evolution, but at the same time, so these artists all work more with very radical methods, actually. So that was my point. If you think that if biologists would agree that this is one line and you can really mix these boundaries.
Well, a good mix, I mean, the Nobel Prize, I think, in biochemistry 5 years ago was dedicated to directed evolution. So it's a genetic modification of bacteria because they have a very fast reproduction cycle, and to actually vary the medium and just not even touch the genetic makeup, but just by an accelerated directed evolution, which is another form of genetic engineering, but without touching the genome as such. So, I mean, the lines are already blurred here at this level. No?
Okay. Okay, we will keep exploring. Thank you. Thank you very much.